the history of Stem Cells Inc. is not the straightforward, plain vanilla history. So Stem Cells, the stem cell biology company, was established in California by three academic gurus in stem cell biology: Irv Weisman, Rusty Gage, David Anderson. And this was in '95, and for about 2 years it was kind of a skunk works project. They looked at all of the various options, including the VCs, in terms of how we're going to fund this endeavor, and decided to merge their company with a publicly traded company called Cytotherapeutics, which was headquartered in Rhode Island. About 2 years after that, the board of Cytotherapeutics decided that their cell encapsulation technology was not as promising as they had hoped it would be. So they sold off that technology, took the proceeds and put it all into stem cell biology, and in the year 2000 they changed the name to Stem Cells Inc. So that's how the stem cell company, if you will, became a public entity. Now, the public entity status, in turn, gave us access to the public markets. I was just looking back. Since I joined the company in January of 2000, we've raised about $186 million, of which about $170 million has been through the sale of equity. Now, what has helped us do that is a combination of a couple of things. Number one, our name. Our ticker symbol is STEM, and our name is Stem Cells Inc. So I think that's an important asset. But more importantly is the quality of the science, the quality of the people who are involved in the endeavor. And in the first instance, in the very early stage of a company where it's all about science, you better have great science and you better have great people. And I think we had those two things going for us. We were able to articulate a relatively straightforward strategy to the marketplace, which was, look, we're not going to swing for the fences our first at bat. We're not going after Alzheimer's or Parkinson's. We're not focused on neurogenesis, creating new functioning neurons. Rather, we're going to look at neuroprotection, if you will, the lower hanging fruit. Not easy, but lower hanging. And so our entire process became focused on demonstrating that we could bring our neural stem cell into the clinic, not do harm to any of these patients, and show that there was a good safety profile on which to then begin expanding our clinical research. So for the CNS program, which is really the major program we're focusing on from a therapeutic perspective, the source of our neural stem cells are from primary tissue, and more specifically what I mean by a primary tissue is from an organ system such as the brain. The neural stem cells that we've taken into the clinic are derived from donated fetal forebrain, and we process the tissue under GTP. It's acquired under GTP, and then we process it under GMP conditions. The process involves dissociating the cells from the tissue, putting them into suspension, and using monoclonal antibodies as cell surface markers to fish out the specific cells that we're interested in. This is called prospective isolation. So we use flow cytometry then to pick out the specific cells of interest, and we reject and leave everything else behind in the population. Typically from one source of tissue, when we're completed with that process, we'll have approximately 1 million cells. And then we put them into a proprietary cell culture system, which further propagates and purifies the cells. So we end up with cryopreserved stem cells in a bottle. It's a variable number, but we can dose many, many patients from one single cell bank. By way of illustration, for the first clinical trial that we completed in NCL, commonly known as Battens Disease, we had 2 dose groups. The first, or the highest group, received up to 1 billion cells directly transplanted into the brain. The lower group received somewhere in the region of about 500 million cells. Now, you wouldn't put that number of cells into the eye or into the spinal cord, so it's a very, very variable answer. The next clinical program, which we recently announced, is in conjunction with UCSF, we're going to initiate a clinical trial in another rare pediatric disease called Pelizaeus-Merzbacher's disease, or PMD. This is a myelination disorder. Some of the pathophysiologies of this condition are common to some forms of cerebral palsy and MS. What motivated us to initiate the study in PMD was, first of all, we had a good safety profile from our first clinical trial in Battens disease. But secondly, we'd shown in an animal model of a hypomyelination model, which is a shiverer mouse, that our cells in graft differentiate to form oligodendrocytes, which are the specialist cells that, if you will, glom on to the axons. And what we saw was that these oligodendrocytes started to weave new layers of myelin. And it was very, very convincing data. We presented the data to the FDA last November, and we got approval within 30 days to initiate the study. So we're moving full steam ahead, and we're now working with UCSF to initiate that study. In the preclinical arena, we've got very strong preclinical packages for spinal cord injury, as well as for retinal degeneration. In both of those areas, we've published preclinical data at the ARVO meeting with regard to the retinal degeneration, and we've also published in peer-reviewed journals the work that was done by our colleagues at UC Irvine on spinal cord injury. So it's very, very exciting stuff that's going on. Our programs now have relevance for the three major components of the central nervous system: the brain, the spinal cord, and the eye. So the near-term value creation milestones that I will be looking for Stem Cells Inc. would be, first of all, enrollment in our PMD study at UCSF, enroll our first patients, dose our first patients. That will be a critical element for us. Secondly, we have a meeting scheduled in November with the FDA to talk about future clinical studies in Battens disease. And depending upon that outcome, then we would hope to initiate another study in Battens disease as yet another step on our way to getting a BLA to market the cells for NCL and Battens disease. Another major thing for us, I think, going forward in the near term will be to start our clinical trial in Belgium, transplanting our HLEC cells into children who've got a variety of metabolic disorders. But we're really looking forward to an exciting 5 years and tremendous progress in the field. It hasn't been an easy task. You know, the pioneers are always referred to as those folks who are lying on the ground with the arrows sticking out of their backs. So, you know, we've taken our lumps. But at the end of the day, you know, it's not just writing the protocol. There's a whole series of checkpoints that you have to go through. And, you know, just getting FDA approval in and of itself is a very, very significant event. But what you then have to do is to get many other approvals. And you cannot initiate a clinical trial in the United States unless you have an ethics committee or an institutional review board approval. So you need that. You need the approval of the clinicians. I mean, they have to buy into this. And in our case, it's not just the neurosurgeons, it's an entire clinical team that has to surround these patients. You also need the approval of the parents and the patients or their guardians. And then you need the approval of the lawyers with regard to the contracts that you have to enter into, and you also need the approval of the finance folks who are going to negotiate very hard on the contract. So it is a long, hard slog to get through the FDA, but then, quite frankly, you have another series of slogs on your hands. So it's a tough process. We've done it. We've managed it successfully now on 2 of our clinical programs with our neural stem cell. And we've recently announced that we've got ethics committee approval in Belgium at UCL Saint-Luc to initiate a clinical trial there in a metabolic disorder, pediatric metabolic disorder, using a cell that was discovered by our scientists at the company. We call it the human liver engrafting cell, or HLEC. So we're right on the cusp of opening up another clinical endeavor, this time focused on liver disease. So we've gained a lot of experience. We have the cuts and bruises to show for it. But all around, it's been a very, very motivating, uplifting experience, and we're very pleased with our progress to date.